The overall goal of this laser capture microdissection protocol is to isolate total RNA from pure populations of prostatic epithelial cell types from heterogeneous frozen prostate tissues for downstream gene expression analyses.
This method can help analyze patient specimens and answer key questions in the prostate cancer research field, such as how RNA expression differs in benign and diseased epithelial prostatic tissue samples or in clinical trial specimens.
The main advantage of this technique is that laser capture microdissection enables RNA isolation of homogeneous populations of epithelial or stromal cells from frozen prostate tissue.
Generally, individuals new to this method will struggle to acquire high-quality RNA because they do not know which steps are critical to maintain RNase-free conditions.
Begin by gathering all reagents and tools needed for the procedure. After allowing the frozen prostate tissue to equilibrate inside the cryostat for 30 minutes, squirt mounting medium into the bottom of the cassette and quickly mount the tissue using chilled forceps. Place the cassette into the chiller of the cryostat for 5 minutes to set the mounting medium. Once set, squirt frozen mounting medium onto the chuck and carefully press the mounted tissue into it bottom side up. Allow the preparation to set for 5 minutes before placing the chuck into the holder. Lock the handle into position and insert a new blade onto the cryostat. After unlocking the handle, carefully advance the sample until the desired tissue exposure is reached. When ready, adjust the cryostat to 5 microns and cut 1 or 2 sections. Place these sections onto a charged glass slide for hematoxylin and eosin.